Okay. All right, we'll get started. So tonight is just going to be about membrane permeability. The last stream was all just about the structure of the cell membrane. And today we're going to talk about how that structure allows for its permeability, which is about what it lets in or out of the cell. So here's our other AP biology stream for tonight. If you haven't already, go ahead and follow Think Fiveable on social media, and also me. I'm not on really Instagram, but I am on Twitter for Fiveable. So at Mrs. Parnan, if you want to follow me there. I'm trying to become more active on Twitter because I know it's a really great place and a lot of good resources are shared. But oh, Dakota, you're feeling bad about this week. I'm sorry. That's no good. Hopefully things turn around. Okay, so if you have not been to my streams before, every now and then I like to just go over some learning tips in AP biology. And I want to just remind you that even though you're taking a very rigorous, very difficult college level course, focus more on learning rather than getting assignments done or preparing for a test or even thinking about the AP test. Focus on every day of just learn, get this information into your brain. If it is difficult, that's actually a good thing. That means your brain is making connections, and neurons, so the cells that are in your brain, they're making new connections too. So I just want you to know learning is not supposed to be easy. If it was really easy, then you're not in the right class. Okay, so just be encouraged by that. If things are too easy, you're in the wrong class. Okay, if things are really difficult, that means your brain is learning something completely new. Okay, so just be encouraged by that. Other ways that you can help yourself with the learning is just keeping up with the assigned reading. Okay, if we're not reading the text, if we're not diving into stuff that we should know, and we're falling behind, then it's going to make learning more difficult. So do your part to make the learning happen. Other ways you could do that is summarize your reading rather than just having a notebook out and taking notes as you read. I would read a little section, like cover the page and summarize what did you just read? What was something that you think is important there, right? It makes you a little bit more of an active participant in your readings and in your note taking rather than just like look at the bold word, copy it down. Look at the example, copy it down. So that's not going to be your best way of doing it. Ideally, you read a little bit. Can you summarize it or even just talk it out? Maybe you're reading with someone and taking turns and you read something about osmosis. Verbalize it to someone and say like, okay, well, I just read about a hypertonic solution. Here's what a hypertonic solution is, or I think, right? And then you might have to go back and check, but those are really good learning habits. Teaching each other, I wrote in bold because I just think that's really great. Form a study group. I know a couple of you who come on here are in class together. So I think that's a huge step, like tell the people in your class about Fiveable and keep each other accountable to go and watch the live streams, or maybe take turns watching the live streams and say like, hey, I watched this really good one. Let's look at the replay and things like that. Learning horizontal. So that's like keeping up with your assigned readings, participating in school. It's all the stuff that is laid out in front of you, like horizontal, left to right, we're reading, we're thinking. But then I want you to think vertically, which means ask questions. So don't just like try to memorize everything you're learning in AP biology. For those of you who just came in, I'm just going over some learning tips before we dive into the content of cell permeability. And we're on this bullet point here about learning horizontal, but thinking vertically. So horizontal learning is like looking at what's already been discovered, right? We're reading in a textbook and we're discussing lab data and everything is left to right. We're reading it on our page. But then thinking vertically is asking a bigger question. So like later today when we're talking about cell permeability and the importance of cholesterol in the cell membrane and what that does for permeability, think about an example of when cholesterol is low, like what would happen if, and so that's thinking vertically of a lot of the times in biology, I always like to think like, well, what if that component stopped working, right? Like if you're in cell respiration and all of a sudden there's no oxygen, what happens? Where is it going to get held up? What's going on here? And so testing yourself in that way is really good too by asking those questions. And then, rewatching streams is always really good. And then talking out the content and then draw it out. I'm a huge encourager of drawing in biology. I think drawing things out is super huge. Even if you don't think you're a visual person, or if you're not great at drawing, I'm not great at it either, but I learn a lot by drawing and making sense of what I've learned on paper. So anyway, those are just some of my tips. And I also want to give you, okay. So here are the objectives for cell membranes. This is what AP College Board would like you to know. So there's a bunch of things there. It's kind of wordy, but I've bolded all the stuff that we're going to go through today. We'll look at structure, fluid mosaic model, what's allowed in and out, and then a brief thing on cell walls. All right, so here's where we're going today. Maybe diagramming. I do have some practice for your responses as well, some FRQs, because hopefully you know now, the AP biology test is 50% a free response question based, okay? So I know it's different than other AP classes, but for AP biology, 50% of your AP score is from the free response and 50% is from multiple choice. And so I like to give my students as much practice with free response as possible, even if it's just a small component, but just getting that practice of putting on paper in your own words, what do you know about this? How could you describe what's going on here? So we'll get to that today too. All right. So just as a review, cells, right? This whole unit is about cells. And if we can keep track and keep grounded in our heads, what is the point of a cell? What do they need, right? We know that we're made up of cells, right? And all of these certain cells work together to form tissues, right? So like your muscle cells all form your muscle tissues, and your cardiac cells form the muscle of your heart and things like that, right? So they obviously have a larger picture in terms of how they help us, but if we think of them individually, what do they need by themselves? So individually, cells need to get necessary resources, right? So they need sugar, or they need, nutrients, okay, to make ATP. And if you're in cellular respiration right now, you're probably like, oh, ATP, yes, okay. But if we to remember, ATP is adenosine triphosphate. It's this energy molecule. It has 3 phosphate groups. So if you remember your functional groups from Unit 1, it's a phosphate with 4 oxygens around it. And so imagine 3 of those phosphate groups linked together, and it's a high energy molecule that's used for all the cellular work in a living organism. Is a super important molecule. Cells also need to eliminate waste, right? Every cell, it needs good things coming in. It also needs to get rid of waste products. And most importantly, that you don't even think about too much, it needs to be able to exchange chemicals and energy with the environment. So cells need to let things in and let things out for reasons of communication, for reasons of doing cellular work, making proteins, exporting proteins, transporting information, sending signals. Think about, like, you hear your phone ring and you pick it up. That's because of a bunch of signals that have been sent, and because of the permeability of your cell membranes that allowed you to actually pick up your phone and make that message be sent. And we'll get more into that later, not today. All right, so another review, just the cell membrane in general. Remember, it's the membrane that actually covers each cell. It separates in from out, right? It gives us a barrier of inside versus outside. Certain things can pass through more easily. Hopefully, we also remember that it's made up of phospholipids. And then also, like in this picture here, this is a prokaryotic cell. Remember that single cell. Prokaryotic means before nucleus, so there is no nucleus. It's just a nucleoid, which is just the glob of DNA and proteins. But they have cell walls, very similar to how plants have cell walls. Okay, so we'll talk a little bit about cell walls too today, just so we're not forgetting about them. But we're mostly focusing on cell membranes and permeability. Okay, so before, I don't want to forget about cell walls, I'm just going to talk about them right now. This is a review from Unit 1, where we talked about macromolecules and how, if we think of the monomer of a carbohydrate was, an example of a monomer was glucose, right? And they could stick together in a long chain. One of the macromolecules that can be made is cellulose, and cellulose is found in plant cell walls, fungal cell walls, and bacterial cell walls as well. And they make these really thick, strong fibers. If you imagine, if you've ever taken a bite of like a piece of celery, and you take a bite of celery and it kind of pulls off in strings, and you think about like how hard that is to chew, and the fact that actually our human bodies cannot break apart cellulose. It actually just comes out. It's called roughage, and so it's a good thing to eat. That's why part of eating vegetables is a good thing, not just the nutrients, but that cellulose kind of helps clean out your intestines because it's not being absorbed or broken down. It just has to be moved through. So just a reminder about cell walls in general. Things can go in and out of cell walls. They are semi-permeable, similar to a cell membrane, but their most important thing is for giving plants, fungi, and bacterial cells structure and strength. So if you haven't gotten into osmosis yet, which we will next week, here these cell walls are helping these plant cells maintain some sort of a homeostasis or equilibrium, even when water levels are high or low. So here, like the internal cell membrane has been plasmolized because all this water left a cell. It's a very dehydrated plant cell, but it's not crumbling. So even if the plant cell is dehydrated, it's not going to just crumble into a million pieces. It's still going to look like a plant. All right, but we'll get more into osmosis much later. Okay. So go ahead and, if you can, give me in the text box if we're looking at the structure of a phospholipid bilayer, what do we mean when we say something has a hydrophilic head? We're looking at this bilayer, we're looking at phospholipid. What does a hydrophilic head mean? Hydrophilic would be water loving. So we have these external barriers on our bilipid membrane layer. And then if we said hydrophilic means bonding with water or loving water, what would hydrophobic mean? Just as a review for everyone. If I said something is hydrophobic. Yeah, good. So the word phobic, that's something that I think is in a lot of people's vocabulary when they say, like, I have a phobia of spiders, or I have a phobia of something else. I'm afraid of it, right? So even though those words sound familiar, do not get them mixed up. It'd be a really easy mistake in a free response question to just start writing and talking about hydrophilic things, but then you may accidentally describe something that's hydrophobic. So just be so sure that you know the difference between hydrophobic and hydrophilic, because it'd be really easy to just write hydrophobic, but then talk about hydrophilic things. So yeah, hydrophobic means they are afraid of water. They will not bind with water. If you remember the actual structure of the phospholipid, right? This is, again, Unit 1 review. So this all builds on itself. So again, it's the importance. And that's why I love you guys can go back and watch these streams. Because if this is something where you're like, I don't remember learning about phospholipids, go back to a stream or go back to your textbook when we talked about macromolecules and what their structure comes from, because understanding the biochemistry will help you understand the functionality of it. So like here is my phosphate head. It's a head because it kind of shapes into this circle. It's hydrophilic, it's charged, it likes water. Remember, again, you got to know water's properties. The fact that water is polar, it's got unevenly charged ends, right? So partially negative, partially positive. And so if I put something negative near it, that's going to be hydrophilic. It's going to want to be near water. Where these fatty acid tails, they're just a bunch of carbons and hydrogens, right? So they want nothing to do with water. You think about oils, you guys gave that as an example when we talked about lipids, right? So the phospho is the top part, that's hydrophilic, it loves water. The lipid part, a lot of you were talking about like oils and fats. That should be something that stands out to you as an example of, okay, well, it makes sense that it's going to be hydrophobic, because I know when I have like a salad dressing, for example, the oil and water components separate on their own. They don't want to be near each other. It's not going to be soluble in water. Okay, so that's why this bilayer forms, because we have these tails, the fatty acid tails, that are afraid of water, right? And so all these cells are in a water environment. That's the other thing too that people forget. And they're when they're walking themselves through a free response question or even reading a multiple choice question, they forget what we're talking about here because they're so focused on the details that they forget to zoom out. This is another study tip for you. When you're looking at something that's complex, let yourself zoom out from it and go, what is this? What's the big picture here? Why does this matter? So when you're looking at just this section of a membrane, zoom out from that and realize you're talking about a membrane of just one cell, right? And that one cell lives in a very aqueous environment. So we start to think about that aqueous environment. It's like, oh, okay, well, that makes sense that the stuff that likes water would be on the outside, right? And the stuff that doesn't like water would be in that inner layer. And you need, if you think about like the intracellular layer, the inside of a cell is also very aqueous. It's filled with cytoplasm. Okay, so that's why we need to have a bilayer. We need to have two layers because of the aqueous environment outside of a cell and inside of a cell. Okay, and that's just a picture of oil and water. If for some reason, when I said salad dressing, you were like, I don't know what she's talking about. So that'd be the example of oil being like the fatty acids here, and how it does not want to be near water. Okay, and that will come into play in a little bit when we talk about who's letting what in. Okay, so another thing that you need to be familiar with is the description of the fluid mosaic model of the cell membrane. All right, so a mosaic, if you didn't know, they're very beautiful, right? So a mosaic means it's generally, I think of art when I think of a mosaic, but basically it's a combination of a bunch of different materials and structures and colors, but then they all are put together to form something beautiful. Okay, so it's like a hodgepodge, I guess, if that's another word that maybe you're more familiar with rather than mosaic. But this is a picture of a ceramic mosaic where it's got all these different tiles, different colors and structures to make something together. But with a cell membrane, it's mosaic because it's not just a phospholipid membrane. It's not just a perfectly phospholipids all in a row and they all just stick together and identical in every cell. It's a mosaic because it is filled with a bunch of stuff, right? So if you've seen a picture similar to this, we have tons of different proteins. We've got glycolipids, we have glycoproteins, we have things that let large molecules in and out. We've got cholesterol wedged in between our actual phospholipids, and each of those things is similar, it's like analogous to having a different color on the painting, right? Or using a different color tile. But then when you put it all together, it forms this beautiful structure that allows a cell to do what it can do. Without these things, the cell membranes of all of our cells would be pretty doomed. They'd be really affected by temperature. They wouldn't be very fluid at all, which hence it's called fluid mosaic. So there are things in here that allow for fluidity of a cell. If they were all just blocks in a row, think about the lack of communication that could happen. Think about how rigid every organism would be. Okay. So if for the purpose of, if you, I don't know if you like take pictures of certain diagrams as we go, I will post this Google slide to Fiveable tomorrow once everything's downloaded and put on the internet. And it has my notes of like what A through G stand for. But if you're wanting to just write it down right now, A is a glycoprotein. So it's attached to a protein that's embedded in the cell membrane and has glyco and protein. So what two macromolecules is that made up of? What two macromolecules is a glycoprotein made up of? Go ahead and type it in. So for A, I've got glyco. I'll type it in here. So we have it, glycoprotein. So what are my two macromolecules that make up that structure? Nice. So carbohydrate, and you could have just said protein, but yes, there's amino acids, of course. The amino acid is the monomer to our protein. So yeah, it's carbohydrate and amino acid. Now B, it looks the same, but they're colored here in green. It's just a model. That's a glycolipid. So glycolipid, right? Glyco, meaning think glucose, right? Carbohydrate. It's a carbohydrate and a lipid together. Okay. Those two things, I like to, because they're hanging like on the top or the outside of a cell, those are huge for cell communication and cell name tagging in a way. So if we think about, if you guys have done blood typing yet in your classes, which I don't normally do until we get into genetics, which might even be in the winter, but we think about blood typing, and you go give blood and they tell you you're type A or type B, type O, or they talk about the importance of donating blood in general because we have different types, right? So only like I'm type B, so I cannot receive from type A, right? If I needed blood, I would be in big trouble if I didn't have a type B blood donor or a type O blood donor, which is the universal donor. But a huge reason for that is that because our blood cells have these name tags on the outside. And so that's what I like to think of glycoproteins and glycolipids as. They can serve as name tags, which is basically like cellular recognition of if something comes into your body and your immune system doesn't recognize the tags that are on the outside of a cell, they will launch an immune system attack. All right. But luckily for us, we have, unless you have an autoimmune disorder, disease, your body is really good at recognizing self, which means if something is, like my body should not attack my intestinal cells, right? Because they are mine. They are my intestinal cells. They're not a donor from anyone else. I shouldn't be attacking my own body. But that's where autoimmune disorders come in, where something is wrong with the cell-to-cell recognition of, like if I were to put another picture here of a cell next to this one, those two cells should be able to recognize each other as self, we say, which means it's not an intruder, and an immune system response does not need to go haywire and cause issues. Same thing with like allergies, right? So like if someone's allergic to pollen, it's because their cells are very, the cellular markers kind of freak out in a way when it's interacting with cell-to-cell recognition. But it's not just about recognition. Glycoproteins and glycolipids are really good at in terms of attachments. And so when we think about, when you guys get into more communication, there'll be another stream on this, but there's this thing called a signal transduction pathway, where basically something that can't get through the cell, maybe it's too big, or maybe it's just, if it's like a steroid that's not going to go through the cell, it can bind to one of these glycolipids or glycoproteins, and then that will cause an internal cellular change that will begin a cascade of events that will either like tell the cell to start making a certain protein or do something with DNA. So anyway, that's what those two things do, A and B. C is a peripheral protein. If you think about what the word periphery means, it means like external, or when you think about like your peripheral vision, it's your vision off to the side, okay? So it's like on the side of the cell membrane here. So C is a peripheral protein. That's another huge one for cell communication and transduction of messages. So something would like bind to the peripheral protein, and then that protein would cause an internal cellular change. So those are all kind of similar in that they're not like all the way through. They're not transmembrane. They're not through the protein. D, letter D, is going to be your integral protein. I'm just going to type it in here, D, integral protein. So that is going to be integrated through the entire cell membrane. The important thing here is to really think about, okay, well, what portions of this protein are going to be hydrophobic and hydrophilic? It's not just going to be this blocked protein that doesn't have to behave according to the biochemistry that we know to be true for a bilipid, a bilayer membrane. So the outer edges of this integral protein are also going to be hydrophilic, and the inside is going to be hydrophobic, just like those fatty acid tails. So that could be, I could see that being like an AP question where they say, we've got this really important integral protein that's supposed to be a channel for such and such amino acid to get through, or protein to get through, but there's a mutation and that makes that protein completely hydrophilic. Okay, and so then you'd have to think about, well, what if the whole protein is hydrophilic? What does that mean? Is that going to cause, a collapse of your membrane because everything on the inside doesn't really want to be near water, and all of a sudden it's letting through all this hydrophilic stuff? And with questions like that, there's a lot of different avenues of being able to answer it. So that's what I mean by starting to think vertically. Almost try to come up with your own questions like, well, what would happen if this were to happen? Okay, E, that is cholesterol in the membrane. Cholesterol is kind of like a unknown hero in a cell membrane, I think, because people don't think about it. Everyone thinks cholesterol is so terrible. When we think of like diets and all that stuff and high cholesterol, it's so bad. But cell membranes really need cholesterol to maintain their fluidity. So we talk about the mosaic of all these different proteins, but the fluidity, a lot of it has to do with, well, saturated and unsaturated fatty acid tails. So those kinks in the hydrophobic tails allows for fluidity. But cholesterol makes sure that the hydrophobic and hydrophilic components are not too close together and they're not too far apart. So without cholesterol, if you had temperature changes that were higher or lower, it would really affect your membrane. And then every time things were hot or cold, that membrane wouldn't be able to function normally. It would either be way too tight and nothing would, small ions that are allowed should just, not small ions, small uncharged things like oxygen, right? It wouldn't be able to go through a membrane when it's supposed to. And conversely, without the cholesterol, at certain temperatures, right? If it was too warm, all of a sudden your pores would open up way too big and things would be let in way too easily. And so cholesterol plays a huge role in both the permeability and the fluidity of your cell membrane. So that'd be a really good AP style question too. Maybe I'm going to write a note to myself to come up with like a free response question having to do with cholesterol. And maybe I'll get some like sample data or something like that to see how we can explain it a little bit better. Okay. F is an example of a channel protein. It's kind of just showing you your integral. Channel protein is an example of an integral protein. So it goes through the whole cell membrane, but it serves as a channel to let things in. So if you've taken AP Psych, how many of you have taken AP Psych already? Give me like a hands up if you've taken AP Psych, because then I can know where we're at in terms of channels. Give me one of these, or just say yes. AP Psych, good. So one of you's taking it right now. Okay, great. Anyone else taking AP Psych? It's a really good class. It's really good. There's a lot of, actually, a lot of my students who have either taken it or take it with AP Biology at the same time, they tend to do really well on both tests. Oh, good. You're taking it next year. So awesome, Maria. Next year when you take AP Psych, a lot of the brain science stuff will be review, and it will just make you soar way higher. You'll be like, oh, I totally know this stuff from AP Biology. I think they're really good courses to go together, or at least take both of them. But anyway, for those of you who have taken it, hopefully you remember when we talk about neurons and we talk about the action potential and how a neuron cell, so a neuron is just the cell of your nervous system. They're called neurons. They're very highly specialized. They're not like these perfect circles. They've got crazy like tree-like parts and then like a tail, and that's just one cell. But anyway, the cell membrane has sodium and potassium channels that are gated, and so they can open and close, which is a whole other thing. Like, we're not even going there today, but the idea of when you have an ion, something that is charged, right? Those fatty acid tails don't like charged things. It's not that they just don't like water. They don't want anything charged near them, right? They're phobic. They don't like it. And so you need these channels to let sodium and potassium through, because that is how this thing called an action potential actually happens in a nerve cell. So if the inside and outside of a nerve cell is not the right charge, right? So like negative or positive, if it's not at the actual right voltage, like even voltage, I think of like voltage from like an outlet that you plug in your iPhone to, right? There needs to be a distribution of charges in order for your phone to actually take on a charge. Your nerve cells are the exact same way. And so F is a really good example of a channel that will just let an ion go through. It kind of protects it so that those tails aren't like blocking it and not letting it through. So for those of you who haven't taken it, you'll learn more about that in AP Bio. This was just like a very brief preview. Anyway, okay, let's move on. So here's just my silly drawing again. I told you I'm not a great artist, but I think drawing is really important. It can help you remember what things are all about. So the function of our membrane, the reason, that it has this mosaic, there's all these different components and it's fluid and that's so great, but its overall function is to be selectively permeable. So hopefully your teacher has broken down that word for you. Think like the word permit, like, oh, I'm going to permit someone to come in and out of the door, right? So like your house, if you have a key or wherever you live, right, a garage code or a key, right, you're not permitted to get inside unless you had a key, right? So like it's selective to who has a key to get in and out of your house or has the code. So permeable, it has the word able in it. So are you able to are you able to go in, right? You have permission to go in. So the membrane is like that, where the actual cell membrane allows certain things to go in and out. That's where the word selectively is as well. Because if we just called it a permeable membrane, that would mean it's permeable to everything. Anything can come in and out. That'd be kind of chaotic. So it's selectively permeable. That means it is selective, that not everything can come through very easily. It doesn't mean nothing can come through if it's, large or small or whatever. It's selective. And so that's why cells have all these other mosaic things like a channel protein or an integral protein or a peripheral protein so that things can get through. So what I want you to do right now, based on my diagram, it's on purpose not labeled super well. Make some observations about what you think some of these things and arrows represent. So I'll give you an example. The positive and negative charged circles have an arrow going toward and then away from the membrane. So to me, that means positive and negative charges are not permitted. Are not permitted to go through on their own. Okay. Ooh, nice. Arthi. Am I saying your name right? Is it Arthi? Yeah, aquaporins are the red part with water. Arthi, okay, great. Yeah, so you already know about aquaporins. Very nice. They've only known about that since the 1990s, so it hasn't been that long. Well, I guess it's been like 20 years, 25 years, but anyway. The blue part is transport through the membrane. Yeah, so good, Maria. So the 3 circles that have the blue line going through it, that's just showing things that can go through the membrane on their own. There's no energy needed. There's no crazy protein needed. They're just going in and out, and that's okay. And there are some things that are allowed just in and out of the membrane. It's no big deal. They're small. They're not polar. They're not disrupting anybody. What about this big pink one? What do we think that's representing? Like a big pink thing. Oh, it could be an organelle. Yeah, that's true. A molecule. Yeah. It does look like an organelle. I probably should have drawn it differently. Yeah, so the pink thing is supposed to represent a large molecule that cannot get through the membrane. So it's got that arrow similar to those positive and negative charges like ions. They can't just go through that membrane, right? Like I drew that big on purpose. It can't just barrel through that membrane and rip it in half. Okay, but if you see an arrow going straight through, that's just going to be passive transport. It doesn't need any kind of help. And by the way, we're going to have a totally separate stream on passive transport, facilitated diffusion, active transport. All those things are coming up as well. So anyway, there's my key. I've got charged molecules. I've got water that can just go through the aquaporins. Small, uncharged, non-polar Again, if you're not polar, you're hydrophobic. So I could have written hydrophobic as well. Hydrophobic, small hydrophobic things can just go in the membrane straight through. Larger and hydrophilic molecules would need help. And so these large ones, they can get through, but they would need a channel protein. They would need an integrated protein all the way through to let them get through. All right. So those are just kind of like the basics. And I think this is the hardest part. If anything, try to redraw something like this for yourself to make sense of who's allowed in and out. Because again, it's very wordy in terms of like, oh, are we charged or not charged? Polar, nonpolar, hydrophilic, hydrophobic. And so many of those things are synonymous, right? So if we were talking about something that's polar, we could also say, okay, well, if it's polar, it's probably going to like water, right? It's going to be hydrophilic. So be aware of those, synonymous characteristics of something. And don't get yourself lost in terms of there's too much information. I don't know what's going to be let through, but just kind of, that's why I think drawing and diagramming in your own way really, really helps step up the learning. So anyway, so water is interesting because it's allowed through. It is polar, but it's not technically charged, right? It's not like, it doesn't have a net negative charge. It's net neutral, but it does have hydrophobic and hydrophilic regions. So even without an aquaporin, technically water could leak through the membrane here and there. But once you get into osmosis and you learn about like, hey, I've got a cell that's in a really salty solution, what's going to happen? Like, how is it going to maintain equilibrium? Is water going to flush in or flush out? We talk all about that in water movement. Those aquaporins are going to be really important because that is how water can be flushed in or flushed out of a cell in order to maintain homeostasis. And here's just a look at what a aquaporin would look like. So you've got your phospholipid bilayer there, and then here we've got our ability of this water channel to be formed. So water can go in and it's not disrupting your membrane at all. Okay, so just to summarize, again, we're almost done here. Small non-polar molecules can move freely. So I didn't give you an example of that yet. So things like nitrogen, oxygen, carbon. So think about like if you're a red blood cell, red blood cells need to be permeable to oxygen and carbon dioxide, right? So if you've got a red blood cell that's full of oxygen, it'd be a huge pain if it needed to, be regulated in a way of allowing oxygen to leave a red blood cell. It needs to just be able to diffuse from high concentration to low concentration. We'll have a whole stream on diffusion as well. And actually, I'm going to write that down too, because I have a good red blood cell affinity for oxygen data set that we could look at that I really like. And I think it'd be helpful. Anyway, okay. So who needs help moving? Hydrophilic things, so things that really like water, right? And you may think like, well, the hydrophobic heads, they're hydrophilic. Why can't we let something through? But remember, those tails on the inside, they're like the warriors, like the barrier of what's letting things in and out. So because of them, because of those fatty acid tails, hydrophilic things need help moving. So like large polar molecules, but even small ions. So those ions would disrupt your hydrophobic tails. Okay. Exceptions to the rule would be something that's small, uncharged, and polar, like water, right? So it can kind of go in and out, but it's going to be super slow unless you have a channel protein. All right. So true or false? Just one quick question, and then we'll do one for your response. Small hydrophobic molecules require embedded transport proteins to dissolve across the lipid region of the cell membrane. So go ahead and just type in true or false. Small hydrophobic molecules require transport proteins to dissolve across the lipid region. Again, so much of this just rests in how do you feel about these terms, hydrophobic, hydrophilic, and like really knowing that cell membrane. So it's not a memorization thing, it's a thinking thing and a learning thing. Woo, excellent. We're all saying false. Good. Yeah, it's false. If you're small, number one, that's a big thing. It's small and it's hydrophobic. And you think, okay, well, the lipid region is also hydrophobic. So it's gonna totally let it fly right through, like allows like. So false, very good. Okay, so here's an example of a free response question. I used to give it as a quiz a long time ago, but thought I'd share it with all of you. So the cell membrane is much more than a passive barrier of the cell. Brainstorm your ideas to answer the questions below in order to show your understanding of cell membrane permeability. So part A is describe in detail the fluid mosaic model of the cell membrane. So that first part, it's very vague on purpose. You may potentially think, what am I supposed to talk about? If you're given something that broad, pick main important things, right? So like, what makes it fluid? What makes it mosaic, right? Like, what are the things I could talk about? Obviously talk about the lipid bilayer. What is that? What's the structure? Maybe talk about cholesterol, and definitely talk about proteins, right? Like those would be like your three big things of a cell membrane is you've got cholesterol, phospholipids, and proteins, and it creates this amazing function. B, for each structure of the cell membrane, explain how the structure allows for the specific function it has. And so I'll give you an example of that. If I'm going to talk about the phospholipid bilayer structure, right, that's a structure, explaining how the structure gives it its function would be breaking down the structure. So you say, well, here's the structure. I've got this hydrophilic phosphate head. Explain what hydrophilic is, right? You'd say hydrophilic means water loving. It means you've got it's this phosphate head. I mean, maybe even draw a picture of the phosphate group if you remember it, and talk about how it's negative and what that means for polarity and interaction with water. Then the lipid section of a phospholipid, talk about that structure and how it's hydrophobic. And then the idea is you link both of those structures and description of those structures too. It's because of this, that idea of you're partly hydrophobic, partly hydrophilic is why the cell membrane forms itself into a double layer, right? Where we get this barrier on the inside and then this double layer of heads on the outside that allows for outer environment of water and inner environment of water. So I know that was a lot of words explaining it, but you'll see that type of question a lot where it wants you to link structure and function. So always just think, okay, well, what's its function? Okay, I'm going to explain the function and then explain how each specific structure aligns to that function. Okay, so if you were jotting down some ideas of how you would answer this, part A, remember I asked to talk about describe the fluid mosaic model, right? So what makes it mosaic? It's got a bunch of different things. I've got some ideas here. Talk about phospholipids, talk about cholesterol, proteins, what are their jobs, glycolipids. Honestly, you could probably pick 3 of those things and dive into them. And then for each of those things, obviously talk about its function as well. Okay, and again, I'll post this. Usually it takes until tomorrow to download the full video, but I'll post the Google Slides for you on the Permeable website. Okay, all right, here's another example of a free response. We've only got 2 minutes left, but this is kind of what I alluded to earlier. But if a cell had a membrane that only consisted of phospholipids, describe how that would influence transportation of materials into and out of the cell. Okay, so think of a cell membrane that only consisted of phospholipids. That is it. Describe how that would influence transportation of materials in and out of the cell. Okay, so can I have someone, glucose could never get in? Yeah, absolutely. Yeah, glucose couldn't get in. If it was just phospholipids, the only thing that could get in were teeny tiny uncharged molecules. Good. Absolutely. If you haven't gotten into like exocytosis and endocytosis and cell-mediated, I can't think of the word. Something, I can't think of it. I told you guys I was so tired. But yeah, we wouldn't be able to export anything either, right? So some things couldn't get in, but we probably wouldn't be able to export anything. If it was literally only a cell membrane and that is it, You could talk about this. You could write a whole paper on this, to be honest. Maybe I'll do that with my students next year, have them write a whole paper. But you could really write an entire 3-page paper all about this and talk about cholesterol. You could talk for half a page or more about without cholesterol. Okay, if temperature changes at all, which for most living things, that happens all the time. Think about it in a given day. You wake up in the morning, you're hot. You step out to your bus stop and you're cold. If temperature was something that affected the rigidity of your cells, it would be chaos. Our cells would be rigid and then too fluid and then just no homeostasis at all. You talk about channel proteins. You could talk about how ions couldn't move in and out. You could talk about osmosis and how without aquaporins, the movement of water would be way too slow. So if something got into a really salty environment, it would be pretty much screwed, and it would not be a good thing for the cell at all. So the sky's the limit with this kind of question. Okay. All right. Let's see. I think that's all, guys, everyone. Any questions about cell membrane permeability? Okay. All right. Thanks, everyone, so much for coming. I know it's 50 minutes of your day, but it's really, really good that you're coming on here and getting some practice. So it's good to see some of you again. Tell your friends in AP Bio as well all about this. The streams are free, so hopefully we'll see you soon. All right. Oh, good, Maria. I'm so glad it helped. Thanks for participating. We'll see you guys next time, okay? Have a good night.